Alright, is anybody having trouble with this? Yeah, a little bit.
No. No, okay, you're actually. Oh, did you load again? Remember two. Put our settings in, so like 188. And then type just do that spot. No, it's actually there. Okay, try again. Close it up?
Yeah.
Let's just close it. Let's come back. No, I'm sorry. Yes. Yeah, makes sense. Maximize it later. Pretty cool, huh? And you can see how just the time differences is really, really different.
Alright, is anybody having any more trouble? You guys feel like you're finally on a good track? It's actually working. This is awesome. I'm really excited because last year it took like 2.5 weeks to get to this point. It was pretty crazy. Because we had last year I didn't get the Matlab put on computers, but I just assigned the subscription for the software for $100 as the textbook. So people had to buy it, but then the trouble was, instead I had them download a huge data set that took like a week and a half to download because it was like 50 EEG files. It was just way too much. And it wasn't working for everybody. And so we did away with that this time around, and we're making it much smoother. So this is great. I'm really excited. So, first I want to ask if there's questions. Now, you guys feel pretty good? Is it too intimidating? Yes, so?
What is the actual bin mean?
Oh, that's a really good question. Alex asked this a moment ago too. What does the bin mean? Right? So we have this idea that there's this bin, and we named it, in your case, I called it 188, right? So the idea is, and we'll get to this as the course moves on, but just generically, it's just the name of the condition of the memory experiment that we want to plot, right? So maybe if we're studying memory, you could look at the brain activity for when we remember, right? But there could also be a totally different category of times that you want to know, what does the brain activity look like when we forget? Right? Or how about when we just kind of remember a little bit, but not all of it? Right? Somewhere in the middle. And so when we look at EEG data, right, it kind of looks like this. Right? Because biology is really messy. Brain activity looks like this. Right? And it should. How many of you guys want your brain activity to look so clean it looks like a flat line? No. Yeah, right? Yeah. This is a time when you want it to be messy, okay? So you want there to be lots of different activity going on, trust me. So this might be, let's say, from the start to the end of the whole experiment. Now an experiment might take, I don't know, 1 hour. Okay? So this is time point 0, and this could be time point 1 hour. So what a bin is you're going to have different stuff happen. Is anybody colorblind and can't see red? Okay, cool. Usually there's about 5 or 10% of the population that can't see red. So I'm going to use a red marker. So within that hour, you're going to have different times than you remember and different times than you forget, right? So you're going to take a snapshot. These are all times when we remember in red. Okay? Now there's going to be other times when we forget. Let's have that be a little green box here. Okay, so this is forget, and this is remember. So to solve this question, what is a bin? What we do is we categorize this. I always tell people it's like sorting your socks in the laundry. It's a laundry bin. I'll get your question in just a second. Don't let me forget.
Oh, it's not a question.
Oh, what is it? What you got?
I can't see it, so.
Oh, you can't see. Saul, can you turn on all the lights? Is that better?
Red being colorblind, so.
Oh, you are red, big colored. Okay. Well, I use red and green, so. Okay, we'll do blue. I did ask. I tried. Sorry, man. Can you see those?
Yeah.
Okay, cool. You guys remember your rods and cone cells from your biopsych and behavioral neuroscience classes? Yeah, pretty cool, huh? So anyways, this time, these boxes might be when you remember, these remember, and it's like kind of like sorting your socks in the laundry. You might put all your dress socks in one bin, all your white tube socks for sports in the other bin, or your striped socks versus your, I don't know, non-striped socks. I don't know. We're at different laundry bins for different categories. And we want to put all the times you remember into one category or bin, and all the times you forget into another category or bin. Now what you guys are plotting on your 3D Scout topography maps, right, that's what we're going to call them, Scout topography maps. Topography means, or we'll call them topo, which is short for topography, which means essentially the shape of that voltage, right? And you'll see that on the colors, essentially. Where is it maximal peak, the red, or maximum trough, the blue? So you might, in the maps that you're plotting, subtract the times you remember minus the times you forget, and that'll be a third bit, which will be the difference between remembering and forgetting, which is what we care about scientifically. What's different in the brain when you remember versus forget? Well, you know the answer to that because you're looking at it on your screen. If you're where there's those big red differences, are where the brain activity is different when you're remembering versus when you're forgetting. For example, does that help, Sal? Yeah? Okay, well, we've loaded a lot into these conversations. So the class today is not meant to get into that granularity of detail. I wanted you guys to see where we're going. I want you guys to just play around and kind of see how this can be kind of fun to see some cool brain activity. And we're going to see how we can tie together this brain activity with different types of memory and different types of failures for memory and attention and brain states and consciousness and all sorts of really cool stuff. And it's going to happen through the magic of Matlab and this GUI that we're becoming familiar with. You guys have done a really, really good job staying together as a team. You guys are really helping each other out. That's really awesome. And also sort of picking up that challenge I dropped earlier in the class to stay patient and not panic and work carefully through these solutions. And we're going to find some new ways to do it because we are exploring. Nobody has ever done what you guys are doing in class right now. Nobody has ever tried to do this with new Matlab installed on these particular computers at CSUSB because we are innovating. It's a really modern class. There's no other class like this in the country. I invented it. It's true. So part of exploration means every now and then you're trying to cut a path through the jungle and then you take a step and you're like, oh my God, I'm in the swamp. Oh, how do I get my foot out of that quicksand? You pull it back and you go walk this other way instead. So we'll have some mix up a little bit here, but you guys are doing a great job. So thank you for your patience. Hopefully you can see that it does work out. There's no reason to freak out because it will work out. And so yeah, that's that. Now, Saul, you asked an earlier question about some of the stuff we were doing on the mission in the summer, and I mentioned that we were doing EEG in there. You guys want to see any pictures from the NASA mission with the EEG? Yeah? Let's see if I can bring one up here. You saw you want to hit those lights? So, this is actually doing EEG in the space capsule. Oh, so I wasn't in space. She was really, really important. Point of clarity.
Well, where was this at?
This was at Johnson Space Center on the ground of planet Earth. But before we send somebody up to space, we're beta testing. And so we're testing out the systems and the technology and the equipment that people will use in space. But if I'm going to send you to space, and I'm going to give you a bunch of jobs to do and technology to use, I want to make sure I tested it out on some other guinea pig on ground so it doesn't goof up on you in space or create a threat or a problem or a danger. And so we do a lot of beta testing. So I was locked up on Earth. You're confined, no internet, no Snapchats, no Instagrams. It was hard. But Earth, Johnson Space Center in Houston. And it was supposed to be a 45-day mission, and it got shut down on an emergency abort and evacuation on day 23 because it turned out we were kept kind of in the dark from the interwebs because we were supposed to be on a deep space mission. So we only had a few inklings of ideas, but we got told that on the emergency radio at like 8 in the morning one day, the mission's been aborted you guys have 15 minutes to pack everything you own and get out of the capsule right now. We've been in the middle of a massive hurricane named Harvey, and it's tornadoes all night, and the mission's canceled. Get out now. And so, you know, there's nothing fake about a real emergency. And we instantly had one, and, you know, we followed directions, got our stuff together, didn't panic, stayed patient, just got our stuff, and came out, and we found a city apocalyptically underwater. And really tragically, and the people trying to get us out of the capsule had their cars swamped just trying to reach us. We were fine, we were physically safe, but they were in danger getting to us. And so, it lasted 23 days. And so anyways, this is in the capsule, this is part of where the flight suit, this is by kind of the medical station. So this is an EEG cap. It looks very, very similar to what we have here at CSUSB in Matlab, which we're going to see. So we're using the same type of equipment that they were using at NASA. Let me see. I might have. Let's see here. I think I should have a video in here somewhere. Is it? Stop me if you guys see anything you want to see, but sorry.
Wilson.
Huh? Yeah, Wilson, there he is. We were locked up. Yeah. You start to get a little crazy after a while. You know? But yeah, this is reminds us of the beach. And, you know, go to your happy place. And let's see here. I'm trying to find. So this is my team. Our team. This is Reinhold Popolitis. He runs the Lunar Reconnaissance Orbiter at Arizona State. He's an he's an engineer there, and they make discovery. They run a satellite that flies around the moon or orbits the moon, and they take a bunch of digital photographs, and then he reconstructs them. We talked about data being all ones and zeros, right? So he takes this massive, high-def digital data of the moon's surface, of all ones and zeros. He does all this crazy math, and he figures out he got a discovery about the moon's surface while sitting in Tempe, Arizona, in the observatory there, that he published in like Science or Nature, which is like one of the biggest journals in the world. And it turns out what they discovered is there's, I guess, we're not really at a farming school. You guys probably haven't farmed, have you? No? No families that come from like agricultural Central California? Well, anyways, you till the land, right? And there's turnover. And so there's always thought that the lunar surface had certain turnover. At a couple 100,000 years, the asteroids keep hitting and you turn over the surface, which we're curious about because as the surface turns over, we will lose Neil Armstrong's footprints, right? And he discovered with all the digital high-def cameras he was taking, or pictures he was taking with the camera, that turnover was happening much faster than we had thought. And so he had a really, really cool paper of discovery. But out of ones and zeros, by the way, right? Again, we could reconstruct those images of the moon's surface the way we reconstruct our images of our brain activity with this software of ones and zeros. This is Dr. Shelley Cazares. She's an engineer from the Pentagon, Oxford, and MIT. She's also from here, I want to say maybe Tehachapi. She grew up right outside of, outside of Bakersfield. I might have the town wrong. But outside of Bakersfield, her family's from out here in California. That's Dr. Paul Hagen. He was the commander of our mission. Awesome guy. He's an engineer at NASA as well. I'm trying to find some EEG stuff for you. Hold on here. Oh yeah, so that's Paul. There. And this is me and Shelley doing some EEG there. And that's Paul with some other forms of EEG. And what we would do is kind of set it up like that. And we would set it up so that, well, this is some virtual reality we're doing. And, oh, you guys want to see some fun? They gave us this shirt that had an EEG cap on it. I told you guys I was a big nerd, right? So, got to kind of dork out a little bit. And that was really fun for me because everybody else is engineers, as you can see. And I actually grew up for the last 15, almost 20 years doing all this EEG. I'm like, oh my gosh, I can't believe I get to do this, and they give me a t-shirt with it. It was really fun. So we got to, cheese it up a little bit. But let me see, there are some really good videos. Oh, here's some other EEG. This is getting ready to go. It's kind of like the lunar surface. They just paint the walls, you know. So we were practicing here, and you could do EEG during physical activity, exercise. It's one of the advantages of EEG over fMRI. So we did that, and there's some more EEG. Ooh, do you guys know who this is? Probably not. This is a Soviet cosmonaut, Andrei Borochenko. And he was just back from the International Space Station. We bumped into him in our hotel, and we colluded with the Russians, and we made friends, and we traded our mission patches. And this is his patch from space. It's really cool. Anyways, look, I was trying to see. Oh yeah, here's some more brain activity you can record. This is called functional near-infrared spectroscopy, and it's a way to measure blood flow in the brain, particularly the frontal cortex, and you have these EEG electrodes on the frontal sites as well. Now, it's interesting to measure this activity because of what we're doing here. Okay, now what this is the exact replica of the system to control the robotic arm on the International Space Station, the Canada arm. And so it's really important to operate that because that's what grasps and locks your resupply vessel with food, water, love letters from your special someone. Okay? And if you can't grab that with the robotic arm, it goes woof. Yeah, it's a long trip. So you really need to get this right. It's very precise, and this is what you're this is a simulator for that, and you don't always get it, and they want to understand how do we learn, how do we make this, what happens in the brain when we're making mistakes, when we're focused, when we're doing it well, and all those kind of elements. Because maybe we can find a biomarker that will send a little alarm off right before you grab that thing about to make an error. Because you can say, no, 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 don't grab it now, you're about to make an error. Save that reach for a moment or two later, and maybe you can settle it in. Make sense? No. I do have, let me see here. Ooh, you guys like MRI? That is going into the MRI, and you can't see it very well here, but this is the MRI machine, and I got to do the MRI while I was wearing my Mars socks. It's kind of cool. Yeah, it's a pretty big dork. It's okay, I got them on now too. Same ones. That's kind of meta, right? Yeah. What's that? Yeah, I went there, Alex. Okay, let me see. Any of you guys like genetics in here? Genes? Biologists? Yeah? So this is mini PCR. You guys know what polymerase chain reaction is when you're trying to replicate DNA? That's what we're doing here. This is a mini PCR machine, and we're getting trained during our training to collect DNA samples and then to replicate it using PCR. And that's what Reinhold is doing here in this Petri dish. And then she's instructing Commander Haugen on how to do that, and Shelley's kind of showing Paul as well. And then we have Wilson, you know, he never really contributes much. He's kind of the stoic, strong, silent type. And he's always kind of lazing around in the chair. He can't do any kind of science, unfortunately. But he's a great listener, you know, he could do it. Anyways, so that's our team, and if I can find this EEG video, I will try to set it up for you. Let me see here. See something cool. This is actually a message to you guys. Let's see if I can get this sound set up.
Hey guys, this is Dr. Addante. I'm sorry that I'm unable to be there the first couple days of class. I've been detained, and in fact detained for 45 days, locked up in a simulated space capsule on a simulated mission to an asteroid. And we're down here at NASA Johnson Space Center on something called the HERA mission. So HERA stands for Human Exploration Research Analogs. And I know I'm scheduled to be there for you guys this fall. I will be there just a little bit late, maybe a week or so.
This is before the hurricane. We didn't know the hurricane was
Dr.
coming.
Amadeo sent in my best, but I wanted to send you my best and let you know that we will be going full tilt once we get back. We're gonna have a lot of fun this semester. We got some really cool stuff to learn about biological psychology. Hopefully you guys are all excited to be beginning your master's degree programs. I'm excited to be there and share that time with you. All sorts of cool stuff about the mind, the brain, behavior, and biological bases for that. We'll cover some learning and memory. We'll cover some brainwave activity, just like we get to record here, and get to have a smorgasbord of science in our class as much as we do here at ERA. And I want to wish you well, let you know that you're definitely on the radar. I'll be seeing you guys soon, but not forgotten you guys, representing you guys with Cal Sticks and Bernardino. Got the Coyotes going, so you're on my mind. I'll see you soon. Keep up with the assignments. The syllabus is accurate. It is expected to be ready to go. And I can't wait to see you guys. Have a great fall. Welcome to Cal State.
Alright, so anyways, that's a little bit of the NASA thing for you guys. Doing the same stuff that we're doing here and getting these same kind of brain signals, same equipment, and we'll kind of teach you guys how to use some of that equipment through the course of this class, which I'm really excited about, as you can tell. And hopefully you guys are, and if you're not, hopefully you'll grow to be excited. If you can't grow to be excited, hopefully you can tolerate it, and get you squared away and on the road to graduation. A little bit of hard work and all that. So take some questions, and then what I was hoping to do for a little bit of time is, if you guys want, we can go down to the lab and actually take a look at some of the EEG equipment, and not do a full training session on it, but just kind of do kind of like what we did here, just a little bit of familiarization, just kind of orientation. Does that sound pretty good? Okay, so we'll do that for a little bit and then call it a day. So I should tell you that the lab is pretty small. We will all fit, but we will need to be very comfy with each other. So be nice to each other, and bring your stuff, because we probably won't come back here. Also, if you want to save any of the pictures that you took from the brain activity or anything else, if you want to keep that, make sure you email that to yourself now from the computer, because there's a chance, as we're going to learn, maybe it won't take, maybe we'll have to rebuild it. So do that before you shut down, and then shut down and log off. And then bring your stuff with us, and we will head down there in just a moment. Sound good? Okie doke.
I think we get it animated.
I'm sorry?
I think we get it animated.